So today I would like to go back to predicting the brain and what's next. So there's going to be a series of few presentations about the brain. So I'm David Holtzman, and I'm happy to show you this piece of information today. So I'm currently working at École Normale Supérieure in Paris and spent my time at the University of Cambridge in the Applied Maths department, where I'm also a fellow of the Churchill College. And please also have a look at Bionumerics, which contain many of our codes. So I think over the 500 years, it has been clear that people have been interested in the brain and understand what it does and what can we do to influence or to modify or to cure the brain. And this is illustrated from the Da Vinci. Da Vinci was able to dissect bodies and extract information about the brain, and understood that there was possibly segmentation of the brain in different regions. And I would say after 500 years, now we are able to record the brain, and this is an example of an array of electrodes. You see each electrodes here marked with a disc, and you can have 8 by 8 or more ensemble of electrode. This is located in the hippocampus, allowing to get recording in real time, and as you see here in red, examples of such a recording. And of course, there is another direction which consists in labeling neurons according to their type. So you can have inhibitory type neurons, excitatory and more refined classification, so that you can see which neuron connect to another one. And that's another fantastic direction of research today. This leads to this kind of map that have been already produced more than 10 years ago to see where neurons are connecting from one brain region to another. And I think for 100 years now, it has been clear that we can listen to the brain in real time through a fantastic observation that the brain can emit electrical signal that can be recorded on the scalp, and you see this as a function of time. You see this oscillation, and has been shown by Hans Berger, German scientist, more than 100 years ago, leading to the observation that we can record from the brain directly by putting electrode and use this to get information about signals of the brain. And this is now routinely used in hospital, and this is an example of what people discovered, that you can go from this type of oscillation to spindle oscillation recorded in the occipital lobe when people are closing their eyes. So where are we going now? This has been there for many years, and what is exactly that we humans are observing, are willing to do? So I would say for hundreds of years, people have had the tendency to get abstract, to go away from reality. And the best example are books, where people are told, or they told themselves a story, inventing in their brain what is projected usually today in movies. And this situation has changed after the revolutionary discovery of chips that led to the first television, where we have been attracted by what was projected on TV. Then in the 80s started a new direction where suddenly we could interact directly with what happened behind the screen. And this was the video games popularized. Then in the 90s started the possibility to have computer at school. This led to a new revolution. And in 2000, the prediction of the movie Matrix said that the future would be a civilization where the reality would be computers. But let's move on to 2010, where the telephone became accessible to everybody, so we could call with the cellular phone from every places. And then with the iPhone, the revolution came that the new reality was what was happening inside the telephone, inside the computers. And so where are we going? So is this the direction inspired by Matrix, that we would live in this matrix, control or not by computers or control or not by algorithm? But how do we go there? Are we ready? And do we have the technology for this? So this is what I would like to say now in the next few slides. So recording the brain. The technology is there. Neuropixel technology allows to have a chip which contains electrodes which can be connected to computers or any device that can receive the signal. So you can inject that directly in the brain, in the different region of the brain, at different locations, and get signals. And so you can see here as a function of time, this is a raster plot indicating here 60 channels, and then the function of time, you see each dot that represent an action potential, and you can see when population of neurons are decharging together. And the difficulty is to make sense of such a plot, to give an interpretation, to understand what it means in terms of neuronal activity and the complex signals, or the complex interpretation, that what it means in terms of thinking or in terms of actions. Here you have another example where you can put the electrodes in a different region of the brain and look as a function of time. This is a raster plot. You can look at how neurons are discharging, and at the same time you can look at the ensemble. If you average this, you get these responses, which is known as local field potential, that give us information about what is the average activity. And so the idea is that one day everything would be able to be analyzed or recorded in real time on your telephone. So the new technology, I would say, is here, but we still need to understand how from this the signal can be reconstructed to get the right interpretation. For example, to move something that is disconnected from us, that would move according to how we would have moved this device had we have direct connection. So basically the idea is that you can inject this in the brain. So this is typically an electrode. This is a convolution of the brain. You need to position the electrode at some place. It's not clear exactly which would be the best position, as we still do not understand specifically what happened in a layer compared to another in terms of signal interpretation. So the channel could be recorded. Of course, it's important to have a low noise so the signal can be extracted. You need to have a good sample rate so that you don't miss information. And everything has to be on a battery that is sufficiently long so that you can get a signal over a long time period. So again, I'm describing Neuralink, but to say that this technology could happen in the next 10 years. But yet a lot of work needs to be done in terms of signal interpretation. So this required to dig a hole in the brain, dig a hole in the scalp, to inject the electrodes. Here you can see examples, and to get recordings. Of course, this requires to dig the hole, but I would say today with what we have seen over the last 20 years, experimentally, once this hole is made, you can put a cover slip that will protect. And this is actually not a real problem to have this little hole in the brain, and people could live with this. So once we have this, we can insert electrodes and get signals. And so the questions that also is important, it's how many neurons can be recorded. So probably hundreds, maybe more, because neurons can go all around. And there's not only neuron, there are other cells. So that an action potential here of the size of the temporal size of a millisecond could be detected. And what we know from literature is that there are neurons that can be discharged for a given visualization of people's portrait. So we know that such a neuron exists. The difficulty, of course, is to find them and to find to whom they are associated with. So this is probably challenging and still to be found, but probably artificial intelligence classification could be used to do that. But it's still unclear how this can be done and how to accumulate the associated databases for each people. So this is probably 10, 20 years or even more. So now once we have this, we need to decode the signal. So from what is taken from the brain, from the implant, to the computer, to the digitalization, and from this digitalization to the ensemble of neuronal activity occurring in different regions, here represented by the spikes of different colors. So this remains to be done, and the technology is not yet there to understand in real time what is this specific type of activity and how can this be translated again into an action given to a connected robots. So there are also, I would say, possible damage to the technology. That is, once the electrode have been implanted, you can see that some types of necrosis appearing. That is a way for the tissue to protect itself. And this is illustrated here, that when after sometimes you implant the electrodes, there is a redistribution of cells, especially microglia, glial cells, that have understood that this is probably a foreign element entering a region that has to be isolated from the rest of the brain. So you have this gliosis activity that could shield the electrode from receiving correct information from neighboring neurons. And thus, this could alter the signal over time. So there are also also understanding that this kind of electrode can also be used to stimulate the brain, leading to the notion that people can lose control of part of their brain, leading to the question of neural rights. And this I would like to refer to Raphael Youssef from Columbia University, who spent a lot of his time trying to see how to protect future generation from, I would say, this kind of possible abuse of the brain. Finally, as we know for decades, is that if you put an electrode in the wrong part of the brain, you can stimulate the dopaminergic pleasure systems that would lead to disconnection of the reality, such as, for example, this rat, although gets a stimulation, a shock stimulation, but by pressing on the pedal, will receive so much pleasure that the rat will forget to eat, to sleep, and can die just pressuring on the pedal. And so this is also something we have to be alert. We have to pay attention for the case where we could have electrode implanted in the brain, and it's a way again to control the people's brain. So in the next lecture, I'll discuss how people have used electroencephalogram signal or local field potential to interpret the signal of the brain from eyes open with rhythm from 15 to 30 or 30 to and more hertz of activity, where this is a different where you have unconsciousness, where you see this slow oscillation at few to 4 hertz and the alpha oscillation from 8 to 12 hertz. And as we deepen into anesthesia, you have this very slow oscillation that can even conduct to what is called burst suppression, where you have a suppression of activity with a burst here, to the extreme where you have flat EEG. So this is just to say that I will come back to this, that after years people have started to understand, for example, the interpretation of EEG in terms of depth of anesthesia. So proposing that in the future we could indeed use our knowledge to interpret the different types of signals. And so to come back to this Neuropixel, I just wanted to show you this quickly, this slide, that if you place the electrode and you record here from the tip, for example, in a region in the cortex, but still with the projection that comes from the thalamus, basically, and I'll come back to that, this is the work of Jeremie Sibillie, that according to the waveform of the recordings, we can distinguish one type of shape, a second type, and even a third type, which allows us to actually make the difference between neurons that are coming from the thalamus versus neurons that are spiking from the cortex. So I'll come back to this, it's very fast, but just remember that the spike shape can be deconvolved, can be isolated, it can be used to detect where the information is coming from. And this is used, for example, here, while we record outside, you have here this local field potential outside neurons. You can see individual neurons and distinguish thalamocortical axon neuron versus the fast spiking in the cortex or regular spiking. So with this, I will stop here to just give you this information that in the future we will be able to have this raster plot to decode the information of the brain, and thus the technology of putting electrodes in the brain will be able to benefit from this decoding. Thank you for your attention, and have a look then at the second video about the brain. Thank you.